second lesson, genetics. We are going to look into genetics lesson. So before we go into genetics, what are the things you have seen in the reproduction? Vegetative reproduction, then sexual reproduction, vegetative reproduction, various types of vegetative reproduction. So vegetative reproduction by root, stem, leaves, and then methods. So in that various types you have seen about it. Then after that, sexual reproduction, structure of anther, structure of ovule, then double fertilization, seed formation. After that, structure of seed. So all these things you have seen in the first one. Now second lesson, genetics. So what is genetics? Genetics is a branch of science which deals with transmission of characters from parents to offsprings. Transmission of characters from parents to offspring. Offsprings in the sense young ones produced by the parents. So that's such young ones are called offsprings. So in that, genetics deals with transmission of characters or traits. Transmission of characters or traits from parents to the offsprings. Actually, genetics deals with heredity and variation. That again you will see into that. Then subdisciplines of genetics. First one about classical genetics or transmission genetics. Deals with transmission of characters from parents to offsprings. Molecular genetics. It deals with the structure and function of genes. It deals with structure and function of genes at molecular level. It deals with structure and function of genes at molecular level. Population genetics. It deals with heredity in groups of individuals or traits which is determined by few genes. Traits which are determined by, but number is given as a E, is which are determined by a few genes. Traits which are determined by few traits which are determined by few genes. Population genetics. Quantitative genetics. Heredity traits deals with heredity of traits. It deals with heredity of traits in groups of individuals where the traits means characters are controlled by many genes. Actually, single character controlled by many genes. For example, skin color. Again, you will get that one. Continuous variation. They will say about that skin color controlled by various types of genes. So like that, even in botany, you just study about the polygenic inheritance like that. So there now you study about that. That's what quantitative genetics. Then now we are going to see about genetics. Actually, genetics deals with heredity and variation. What is the heredity? Heredity is the transmission of characters from parents to offsprings, parents to young ones. And that's what heredity. Heredity deals with transmission of characters from parents to offsprings. Variations. Differences existing among the individuals of an organism. Differences existing among individuals of organism. That's what called variations. So variations are of two types. Continuous variation, discontinuous variation. First one about discontinuous variation. So here under the discontinuous variation, see some characters in a population show a limited form of variation. Some characters in a population show a limited form of variation. Then such characters are determined by a pair or two pairs of genes. A pair or two pairs of genes, and then these characters are specific. Actually, there are no intermediate forms. The characters are not affected by environmental changes. Environmental changes will not cause any effect on characters, environmental changes. And then, now example for this, height of pea plant. Then style length in height of pea plant, style length in Primula chinensis. So these are all discontinuous variation. For example, tall, dwarf. You get tall in the F1 generation. You make a cross between tall and dwarf, you get tall in the F1 generation. Then when you just self-fertilize tall plants, you get only tall in the next tall and the dwarf in the ratio of 3:1. So here and all, you get either tall or dwarf, but not intermediate forms. So that kind of variation, discontinuous variation. So example for discontinuous variation. Discontinuous variation. You just see, well, just give one example. Height of the plant. Tall into dwarf. Cross between tall into dwarf. You get only tall in the F1 generation. When the F1 hybrids are self-pollinated, these are self-pollinated. Self-pollinated, the offspring produced in the F2 generation the ratio of 3 tall is to 1 dwarf. So this kind of inheritance, this kind of variation, discontinuous variation. Then continuous variations due to occurrence of more than 2 genes at the same locus. More than 2 genes at the same locus, in the sense, you just see that here is what chromosome. Capital T, capital T. Capital T, small t, small t. Here you get capital T, then small t. So the point at which gene is located is called locus. So variation due to occurrence of more than 2 genes at the same locus. More than 2 genes at the same locus, in the sense, here 1 gene per chromosome. Here and all we get 1 gene per chromosome. But there, actually in a population you get only alternate forms of characters, either tall or dwarf, nothing else. Whereas here, occurrence of more than two genes at the same locus. Example for blood group inheritance. IA, IB, IO. So like that, these are more than 2 genes located in the same locus. So that is what called continuous variation. So in the continuous occurrence of more than 2 genes at the same locus, occurrence of more than 2 genes at the same locus, how it will be? IA. Then IB, like this. Then OO. So like that. So the same chromosome. For example, any number of chromosomes, 23 pairs of chromosomes. You just take any one of that chromosome that will contain this kind of gene. Of course, more than 2 genes, in the sense you get what, which are the groups from this? A group blood, B group blood, O group. Apart from that, you get AB group also. Okay. So this is the occurrence of more than 2 genes at the same locus in a population. So in that condition, there are intermediate between one extreme to other extreme. There are intermediate between one extreme to other extreme, in the sense there are intermediate. So what kind of intermediates we get, in the sense? See, for example, skin color. Example, skin color in human, height in human, then skin color in human. So these are the example for continuous variation. So here you get dark red, dark black, deep black, dark black, deep black, then intermediate black, then light black, then mulattoes, white like that, mulattoes, albino. So like that you get difference from according to the number of genes, you get this one. So this kind of variation is called continuous variation. So according to the number of genes, you get the character. So that's what continuous variation. There are intermediate characters from one extreme. This is one extreme to other extreme. So that is what the continuous variation. Example, human height and skin color. Then importance of variations. So what for variations? Variations play an important role in evolution. For the original variations play an important role in evolution. How? It plays a significant role in origin of new species. The next one about variations make the individuals better fitted in the struggle for existence. Struggle for existence, you study Darwin's theory of natural selection. So those individuals with useful variations that can be able to survive, and then they are commonly called fittest one. Okay. Then provides genetic material for natural selection. Genetic material for natural selection. Same thing, more or less same. Then consider raw material for evolution, for the origin of new species. And also variations play an important role in high yield, disease resistance. Then some important characters. So in that way, variations play an important role in that. So now you just see that here continuous variation and discontinuous variation. So just like that, you go through the continuous variation example, height in pea plant, then style length in Primula chinensis, where you don't get any intermediate form, but here you get intermediate form. Their variations are due to environment as well as due to genetic level. So here the variations which are due to environment and genetic constitution, genotype. So such variations are called continuous variations. So with they play environmental genetic makeup play an important role in continuous variation. So environment play an environment that can change the genotype of an organism. So that kind of variation is the continuous variation. Now next one about, before you go into in the skin color, you get various types of skin color. The next one about Mendelism. Gregor Johann Mendel, who first conducted experiments on genetics. He conducted experiments in garden pea plant, and then he was the first to publish. He was the first to describe experiments on genetics. So because then experiments made by genetics not disproved so far. That's why Gregor Johann Mendel commonly referred to as father of genetics. And then the term genetics was coined by Bateson. The term genetics was coined by Bateson. The term genetics was coined by Bateson, W. Bateson. Then next one, Darwin's theory of natural selection. Already you studied about that. Now, Mendel's laws of Mendel. Mendel proposed various laws of genetics. Which are the laws? Law of unique characters, law of dominance, law of segregation, law of independent assortment. So, because these laws were not disproved so far. Because of this, Mendel, who is commonly referred to as father of genetics. And then Mendel, biography of Mendel. Mendel was born in the year 1822, July 22nd, 1822, in a small village, Heinzendorf, Silesia, where it is located in Czechoslovakia. Now, actually, after his school studies, he joined in the University of Vienna. He joined the University of Vienna, and then there he studied botany, physics, and mathematics. This knowledge of mathematics helped him to bring out the probability among the species, probability among the individuals in each and every cross. So the knowledge of mathematics helped him to know about the ratios in each and every generation. Okay. Then he joined in a Augustinian monastery at Bren as a priest. He joined as a priest, Augustine monastery at Bren. Then after that, he joined as a teacher of natural science. Along with that, during that time, he conducted experiments on god and pea plants. He conducted experiment on god and pea plant from year 1856 to 1863. So 8 years of experimental work on god and pea plant. And then after that, he just got results of his experiments and then published the results of his experiments in the form of a paper entitled Experiments on Plant Hybrids. Experiments in plant hybridization only. Experiments on plant hybrids in front of Theosophical Society at Bren. In 1866, he just published the paper in Bren Society of Natural History. So that's why History Society of Bren. That time his experiments were not accepted. Why? Because actually he was far ahead of his times. Supposed to be given after a number of years given by him. So he was far ahead of his time. So none of them accepted his views. So his views were accepted only in 1900. Laws of Mendel were accepted and confirmed by accepted and confirmed by three different scientists: Hugo de Vries, von Schermach, and Correns. So they just reconducted experiments on genetics, Mendel's experiments, and then accepted the results of his experiments. So till then, none of them accepted. So, but he died in the year 1884. This was biography of Mendel. Okay. So Mendel, who first conducted experiments on genetics, he was the first to explain about genetics. And then now what happens here? Because of the laws proposed by Mendel, collectively called Mendelism. Okay. Now Mendel selected which plant? Why? Which is responsible for Mendel's success? So Mendel's success is about due to certain things. So which are responsible for Mendel's success in the sense he selected garden pea plants for his experiments. That's all one. The next one, garden pea plant can be grown at any condition. Then mathematical knowledge helped him. His mathematical knowledge helped him to give the ratios of each and every generation. He just conducted large number of experiments on large group of plants, not small groups, large group of plants. Then reasons for Mendel's success. He applied mathematics and statistical methods to biology and laws of probability for his breeding experiments. So that's why now the Bren Society of Natural History, they didn't accept. They thought that mathematics had nothing to do with biology. But he applied mathematics and statistical methods to biology and laws of probability to his breeding experiments. His experiments were carefully planned and he used large samples. The pairs of contrasting characters were controlled by factors, genes present on separate chromosomes. He selected pure breeding as parents and the purity was tested by self-crossing for many generations. So reasons for Mendel's success in this, he just applied mathematics and statistical methods to biology and laws of probability for his breeding experiments. His experiments were carefully planned. He used large samples. The pairs of contrasting characters were controlled by factors. Actually, in garden pigment, there are only 7 pairs of contrasting characters were obtained. 7 pairs that we will see after this. 7 pairs of contrasting characters. Such 7 pairs of contrasting characters controlled by genes located on 7 pairs of chromosomes. In pigment, there are only 14 chromosomes. 7 pairs of chromosomes, 14 chromosomes. So 1 gene per chromosome, 1 factor per chromosome. So then only which law possible? Law of segregation, law of independent assortment are possible, that we will see later. And then he selected few breeds as parents, and the purity was tested by self-crossing for many generations. So what is by self-crossing? Self-crossing in the sense crossing with same character. That's what self-crossing. If you make a cross between tall with tall, you get only tall in the F1 generation. Then when you self-pollinate this one, you get tall in the next generation. So this is what called selfing. Selfing. So whatever may be the number of generations, you get only the same. So that's what called self-pollinating, self-fertilizing one. So you get same character. That's what given there. The selected few breeds as parents, and the purity was tested by self-crossing for many generations. Selfing means again tall with tall. Tall with tall. You get only tall. Again like that, tall with tall. You get only tall for number of generations. So that's what called self-fertilization. Self-fertilizing one, self-pollinating, self-knowledge, selfing, totally called selfing. So what you mean by pure breed is one which can able to give rise to same kind of offspring for many number of generations on self-pollination and self-fertilization. Pure breed is one which can able to give same kind of offspring for many number of generations on self-pollination and self-fertilization. The next one, selection of pea plant by Mendel. Why Mendel selected garden pea plant? This is one success of Mendel. And then selection of pea plant by Mendel reasons. It's an annual plant. Pea plant is an annual plant. So actually it can be grown at any place. And then it completes its life cycle within a short period of time. That's why it's an annual plant with contrasting characters controlled by single gene separately. Single gene separately means 1 gene per chromosome. Single gene separately in the sense 1 gene per chromosome. It's an annual plant with contrasting characters controlled by a single gene separately. Under normal condition, self-fertilization occurred. He used both self-fertilization and cross-fertilization. Actually, it's a self-pollinating one. Self-pollinating one bisexual flower. Flowers are large. You can just conduct artificial cross-pollination. So that's why this plant, it's an annual plant with well-defined characters. It can be crossed with well-defined characters. The characters are controlled by genes located on the chromosome separately. Then next one, under normal conditions, self-pollination occurs. Then you can just made self-fertilization as well as cross-fertilization. So in that way, artificial cross-pollination can be done in that plant. Then the flowers are large. So when flowers are large, self-pollination, but you can just artificial cross-pollination by the removal of stamens. That's what the emasculation. So hence emasculation and pollination are easy for hybridization. Emasculation means the process of removal of stamens from a flower is called emasculation. So emasculation can be done. So this is different, this one different. Selection of pea plant by Mendel. What reasons for selecting pea plants by Mendel? This one reasons for Mendel's success. Somewhat related but different one. Okay. Mendel selected 7 pairs of contrasting characters in pea plants. Such 7 pairs of contrasting characters include height of the plant. Height of the plant in this is tall, dominant 1, dwarf, recessive 1. Then next one about seed form, round seeds, wrinkled seeds, round seeds dominant character, wrinkled seeds recessive character. Then color of the cotyledon, yellow cotyledon dominant 1, green cotyledon recessive 1. Then next one, position of flower, axial flower. Then terminal axial flower dominant, terminal flower recessive 1. Then seed coat color. Seed coat color. So in that seed coat color, green color dominant, yellow color recessive. Seed coat. Yellow color dominant, green color recessive. Then color of pod shape, pod form. So in that pod form means the total fruit, legume fruit pod. So in that inflated means உம்பியது, constricted, சுருங்கியது, seed size, seed form, round seeds, wrinkled seeds. Round seeds dominant, wrinkled seeds recessive one. So like that he just found out 7 pairs of contrasting characters, like 7 pairs of contrasting characters given here. Seven pairs of contrasting characters, stem length, tall, dominant one, dwarf recessive one. Then seed shape, round seeds, wrinkled seeds. Cotyledon color, yellow, green. Flower color, purple flower, dominant over, white flower. Then pod shape, inflated, constricted. Pod color, green, yellow. Flower position, axial, terminal. You just see that here, height of the plant, tall, dominant, dwarf recessive. Then gene, LE. Seed shape, round seed dominant over, recessive one. So here RR. Then cotyledon color, yellow cotyledon, green cotyledon, I. Then flower color, purple color, white color. And then A. Pod shape, inflated, constricted, V. Pod color, green color, dominant, yellow color, recessive. Flower position, axial flower, terminal flower. So what it is, pod means a fruit. It will be like this fruit. This one pod. Then pod shape, pod color. Then flower position means this is axial. Then when it is near the tip, it is terminal. Axial, terminal. These are the 7 pairs of contrasting characters found out by Mendel in garden tree plant. Okay.